Electron transport chain and oxidative phosphorylation. In these videos, we discuss electron transport chain and their complexes, flow of electrons in NADH and FADH, what is ATP synthase and the reasons of ATP synthase, and how to produce ATP by ATP synthase, chemiosmotic theory of oxidative phosphorylation and mechanism of oxidative phosphorylation, how to control respiratory control of oxidative phosphorylation, substrate level phosphorylation, inhibitors of electron transport chain and oxidative phosphorylation, what is uncouplers, and related MCQs in this video. So first we discuss what is electron transport chain. Electron transport system, also known as electron transport chain. In this electron system, there is many complexes which is located in the inner mitochondrial membrane, and these complexes are transfer the electron to the electron donors to the electron acceptor, which facilitate the production of ATP through oxidative phosphorylation. In electron transport chain, there is a many electron carriers which accept the electrons and transfer to the different complexes in electron transport chain. First is FMN, which is known as flavin mononucleotide. It transfers the electron to NADH and reduce to the FMNS2. Second is ubiquinone, which is known as coenzyme Q. It is a fat-soluble quinone and it is a constituent of mitochondrial lipid. And this allows the molecule to move between the flavoprotein and the cytochrome. Third is iron sulfur proteins, and fourth is cytochrome heme protein. Cytochrome heme proteins are many types: cytochrome B, cytochrome C1, cytochrome C, cytochrome A1 and cytochrome A3. Cytochrome A1 and cytochrome A3 are a complex 4 which is a cytochrome oxidase. Structure organization of components in respiratory chain. Components रेस्पिरेटरी चेन में जो कॉम्प्लेक्सेस अरेंज होते हैं वो इंक्रीजिंग ऑर्डर ऑफ रेडॉक्स पोटेंशियल में अरेंज होते हैं. रेड्यूसिंग इक्विवेलेंस फ्लो थ्रू द चेन फ्रॉम द कंपोनेंट्स ऑफ मोर नेगेटिव रेडॉक्स पोटेंशियल टू द more positive redox potential. So in respiratory chain, complex 1 is a negative redox potential and complex 4 has a more positive redox potential. In electron transport chain, our complexes are arranged from more negative potential to the more positive redox potential. In electron transport chain, there is a 4 enzyme complexes which is present and catalyze the transfer of electron through different electron carriers to the molecular oxygen. First is NADH oxidoreductase. This transfer the which is known as complex 1 and is formed by the NADH dehydrogenase and the FES center that catalyze the transfer of T electrons from NADH to ubiquinone. Next is succinate key oxidoreductase. This is a complex 2 which consists succinic dehydrogenase FAD and FES center and catalyze the transfer of electrons from fatty acid and glycerol 3-phosphate to ubiquinone through FAD and FES center. Next is cytochrome C oxidoreductase, which is complex 3, is also known as cytochrome B C1 complex, and it contains cytochrome B, cytochrome C1 and iron sulfur center. Next is cytochrome C oxidase, which is complex 4 and it has 4 redox center: cytochrome A, cytochrome A3, copper A center and copper B center to heme. It involves the transfer of T electron from cytochrome C to the molecular oxygen and forming water. So we discussed electron transport chain in flow of electron. So first NADH, it formed from the PDH complex, T-cell cycle, beta oxidation and malate aspartate shuttle. So from this reaction NADH form which is entered to the complex 1 and electrons are accepted by the FMN and thus electrons are transferred to the FES. Then FES donate the electron to the coenzyme Q and 4 protons are transferred into the inner membrane space. Next, coenzyme Q then transfer the electron to the complex third, which is cytochrome C oxidase, and cytochrome B accept the electron from the coenzyme Q. Whenever electron transport chain begins with NADH, so the order of the complex is complex 1, complex 3 and complex 4, because complex second are related to the FADH. So electrons are transferred to the cytochrome B. Then cytochrome B donate the electron to the FES center, and then FES to say cytochrome C1. Then cytochrome C1 transfer the electron to the cytochrome C, and there is a 4 protons are transferred in the inner membrane space. Then cytochrome C donate the electron to the cytochrome A and cytochrome A3, which is a complex 4. Then cytochrome A transfer the electron to the molecular oxygen, which form the water. Now 2 protons are transferred in the inner membrane space. So there is a total 10 protons present in intermembrane space when electron transport chain are start with NADH. Now discuss FADH electron transport chain. So FADH are formed during the conversion of succinate to fumarate in the citric acid cycle, and FADH complex second accept the electron from the FADH and then transfer the electron to the coenzyme Q. And in this stage there is no transfer of proton in the inner membrane space. Then coenzyme Q transfer the electron to the complex third, which is a cytochrome B, FES and cytochrome C1, and then cytochrome C1 transfer the electron to the cytochrome C, and there is a 4 protons are transferred to the inner membrane space. Then cytochrome C transfer the electron to the complex 4, which is cytochrome A and cytochrome A3. Then cytochrome A3 transfer the electron to the molecular oxygen and form the water. So it is when and 2 protons are transferred in the inner membrane space. So if electron transport chain are start with FADH, so there is a 6 protons are present in the inner membrane space. Then next is complex fifth, which is ATP synthase. So ATP synthase use the proton gradient to form the ATP. So act as a complex fifth, act as a molecular turbine and complex of oxidative phosphorylation, and it utilize the flow of proton down their concentration gradient to generate ATP. And this coupling of proton flow with ATP synthesis is known as oxidative phosphorylation. So protons are entered to the matrix by the ATP synthase, and this protons gradient are used by the ADP and inorganic phosphate to generate the ATP. And this is known as the production of the ATP is known as oxidative phosphorylation. So now we discuss what is ATP synthase. ATP synthase is known as F1 F0 ATPase, and it is the fifth subunit of the oxidative phosphorylation complex. It uses the energy from electrochemical gradient of protons created by the electron transport chain to produce ATP from ADP. So it uses the electrochemical gradient of protons which is created by electron transport chain through the NADH and FADH, and protons are present in inner membrane space which create the proton gradient, and this is used by the ATP synthase to produce ATP. And the structure of ATP synthase consists of 2 main protein components. There is a F0 portion which is attached to the inner mitochondrial membrane, and water soluble F1 portion in the mitochondrial matrix. So it is a ATP synthase, and this region is F1 which is attached to the inner mitochondrial membrane, and F1 portion is present in mitochondrial matrix. So first we discuss about the F0 portion. F region is embedded in the cell membrane and it composes three subunits A, B and C. And F0 region acts as a proton channel allowing the passage of proton across the membrane. So it allows the pass the protons from inner membrane space into matrix. And there is a ring composed of 9 to 12 C subunit. So C subunit has a 9 to 10 ring and arranged in circular way. This ring is tightly linked to A subunit and B subunit of F0. A subunit acts as a mediator of proton transfer between the C ring and the surrounding environment outside the cell. So A subunit acts as a mediator of proton from inner membrane space to the matrix and it allows the pass to proton from inner membrane space to the matrix. Now discuss F1 region of ATP synthase, which is this, and F1 region is a catalytic part of the ATP synthase and it is responsible for the synthesis of ATP using the energy generated by the proton flow through the F0 region. F1 region lies in the mitochondrial matrix and it has 5 subunits: alpha subunit, beta subunit, gamma subunit, delta subunit and epsilon subunit. Alpha and beta subunit consist 3 copies and form the hexamer, and delta, epsilon and gamma has each subunit. 3 copies of subunit alpha and beta combine to form a hexameric ring structure called alpha 3 beta 3 structure, which is main part of involved in the synthesis of ATP. Only the beta subunit are capable of catalyzing the phosphorylation of ADP to produce ATP. So only beta subunit are involved in the production of the ATP, which is a catalytic part of ATP synthase. Oxidative phosphorylation. So ATP synthesis occurs through oxidative phosphorylation and electron move through the chain. Protons are pumped across the inner mitochondrial membrane. This proton movement creates a proton gradient in the inner membrane space, also known as proton motive force, which protons accumulating in the inner membrane space. So during the electron transport chain from the NADH and FADH, there is a creating a proton gradient in the intermembrane space of mitochondria, which form the proton motive force, and this proton motive force help to utilize the protons and form the ATP by the ATP synthase. So by describing this proton motive force and how ATP synthase form the ATP, there is a theory which is known as chemiosmotic model and chemiosmotic theory, which is proposed by the Peter Michel, and explain the how this proton motive force is utilized to synthesize ATP. So there is a lot of protons in intermembrane space which is generated by the electron transport chain, and it creates chemical potential and electrical potential. Chemical potential are generated by the presence of H plus ion in the inner membrane space, then high than the matrix, and electrical potential are generated by the positive ion which is present in inner membrane space is high in comparison to matrix. So the chemical potential and electrical potential help to ATP synthase to generate ATP in the matrix. Efficient ATP synthesis requires adequate supply of ADP to mitochondrial matrix and transport of ATP out of matrix. So this transport of ATP-ADP mediated by ATP-ADP translocase. And the ATP synthesis by the FADH and the NADH is defined by the PO ratio, which is the comparing of amount of ATP produced from the movement of two electrons through an electron transport chain to the amount of oxygen atom reduced to water. So PO ratio is a different for NADH and FADH. It is the comparing of ATP generated through the oxidative phosphorylation and oxygen is reduced by the electron transport chain. So PO ratio is a number of inorganic phosphate molecule utilized for ATP production for every atom of oxygen consumed. So the PO ratio for NADH is 2.8 ATP, 2.5 ATP by the 10 protons. So 10 protons are generated by flow of electron from complex 1 site. Site 2 is flow of electron from complex 3 and third is flow of electron from complex 4. So there is a 10 protons present in the inner membrane space when the electron transport chain are start with NADH. So the PO ratio for NADH is a 2.5 ATP. Next, the PO ratio for FADH2, so it is 1.5 ATP by the 6 proton. So site 1 for flow of electron from complex third because complex second no transfer the any proton in intermembrane space. So 4 protons by the complex third and 2 electrons from protons from the complex 4. So there is a total 6 protons present in inner membrane space which generate 1.5 ATP and the PO ratio for FADH2 is 1.5 ATP. Now we discuss mechanism of ATP synthesis. So H plus enter the intermembrane space into subunit A of F0 region. So F0 region has a subunit A which allow the entering of proton from to ATP synthase and then C ring within F0 rotates. So after the entering of the proton, C ring of F0 rotates allowing proton to move across the membrane. The rotating of the C drive the movement of the gamma subunit in the F1 region, which triggers changes in the conformation of the beta subunit. So alpha subunit allow the passage of proton from enter into the matrix and this entering of the proton rotate the subunit C of F0 region and after the rotating of the subunit C it also rotate the gamma region of F1 region. So gamma when the gamma subunit is rotating, it triggers the conformation change in beta subunit and alpha subunit of F1 region. So conformation change in the beta subunit of F1 region, then rotation of the gamma cause conformational change in alpha 3 beta 3 hexamer of F1 facilitating the binding of ADP and inorganic phosphate to form ATP. So after the rotating of the C unit and the gamma unit, it causes the conformational change in alpha 3 beta 3 hexamer of F1 region which facilitate the binding of ADP and inorganic phosphate to form the ATP by the oxidative phosphorylation. And this process is called rotatory catalysis and it is explained by the binding change mechanism. Binding change mechanism is first proposed by the Boyer suggests that the rotation of subunit within the ATP synthase complex facilitate ATP synthesis. During each 1 degree rotation of the gamma subunit, it contact a different beta subunit causing conformational change in the beta subunit. And the 3 beta subunit can exist in 3 forms. So beta subunit in the ATP synthase exist in 3 forms: O form, L form and T form. The first form of beta subunit is loose form. In this form, ADP and inorganic phosphate are bind with this beta subunit, and then it convert into the tight form, and in the tight form ADP and inorganic phosphate are form the ATP. But in this form of tight, in the tight form beta subunit, ATP are not released. It is only produce ATP but not release. Then beta subunit of ATP synthase convert into the open form, and in this open form ATP are released, and then it also convert in the loose form. Then ADP are bind, and again to again this form is interconvert by the beta subunit. Then the respiratory control of the oxidative phosphorylation. So under physiological condition, electron transport is tightly coupled to oxidative phosphorylation. In the presence of excess substrate and oxygen, respiration continues until all ADP is converted to the ATP. After that, the respiration rate or utilization of oxygen decreases. So if the substrate and oxygen are present in excess, so continuous ATP is synthesized, and after the respiration rate, utilization of oxygen decreases. This means that the presence of adequate oxygen and substrate, ADP becomes rate limiting. It exerts a control over the entire oxidative phosphorylation process. So presence of ADP is required for the oxidative phosphorylation and continuous ATP synthesis. And the regulation of the oxidative phosphorylation by ADP level is called respiratory control. What is substrate level phosphorylation? So in the substrate level phosphorylation, formation of ATP is directly coupled to the metabolic process without involvement of electron transport chain and molecular oxygen. It is called the production of ATP at the substrate level. So in this reaction, when the ATP is generated without entering into the electron transport chain, it is known as the substrate level phosphorylation. And the example of the substrate level phosphorylation is the phosphoenol pyruvate to the pyruvate, 1,3-bis phosphoglycerate to the 3-phosphoglycerate, which is a reaction of the glycolysis, and the succinyl-CoA to the succinate, which is a reaction of the TCA cycle. Next, inhibitors of the ATP synthesis. So there is a three way to inhibit the ATP synthesis. First is the inhibitors of electron transport chain. Second is inhibitor of oxidative phosphorylation. And third is uncouplers of oxidative phosphorylation. First we discuss inhibitor of electron transport chain, which inhibit the flow of electron from the different complexes to the complex 4. So if any inhibitors which inhibit the electron flow from the complex 1 to the coenzyme Q, so that is chloropromazine, retinone, amobarbital and persidine. And some inhibitors inhibit the flow of electron from the complex third between the complex third and cytochrome C, that is BAL, dimercaprol, anthocyanin and naphthoquinone. And some inhibits the succinate flow of electron from succinate to the complex 2, which is malonate, and also inhibit the flow of electron from the complex 2 to coenzyme Q, which is carboxyl. And then some inhibitors inhibit the flow of electron from the cytochrome A3 to molecular oxygen, which is carbon monoxide, cyanide, azide and hydrogen sulfide. Next is inhibitors of oxidative phosphorylation, that is oligomycin, which inhibit the F0 F1 ATPase by blocking proton flow through F0. So if protons not enter in the matrix from the alpha subunit to the beta subunit, so there is no synthesis of ATP by the ATP synthase. Next is orovertin, which inhibits the F1 region of ATP synthase. Then atractyloside, which is inhibit ATP export by inhibiting ATP and ADP translocase. So for the continuous synthesis of ATP, there is requirement of ADP from the cytoplasm and the movement of ATP from matrix to the cytoplasm, which is regulated by the ATP-ADP translocase. So if there is no presence of ADP in the matrix, so no formation of ATP by ATP synthase. Uncouplers of oxidative phosphorylation. Uncoupling agents are lipophilic lipid-soluble compounds which is diffused through the mitochondrial membrane and capable of binding H plus ion. So uncouplers allow the transport of proton across the membrane toward the side with lower H plus ion concentration, thus preventing the formation of proton gradient which is required for the formation of ATP. So in the electron transport chain, there is a proton transfer to the inner membrane space which generate the proton gradient and form the proton motive force. This proton motive force help to ATP synthase to produce ATP. But if there are uncoupling agents which diffuse the proton from the inner membrane space to the matrix, so there is no formation of proton motive force for ATP synthase and no generation of ATP by ATP synthase. So the energy produced by the transport of electron is released as heat rather than being used for synthesis of ATP. The example of uncouplers include 2,4-dinitrophenol and dicomerol as an anticoagulant. Ionophores, they are different from uncoupling agent and they promote the transport of cations other than H plus ion through the membrane and abolish the membrane potential or pH gradient across the membrane, and the phosphorylation is completely inhibited. The example of ionophores is valinomycin and germicidin. The next is thermogenin, which is a natural uncoupler and also known as uncoupling protein, and this protein transport proton back into the matrix without passing through the F0 F1 ATPase complex. So thermogenin is transport the proton from intermembrane space to the matrix without ATP synthase complex. As a result, energy of oxidation is not conserved by ATP formation, but it is dissipated as heat, which is used to maintain the body temperature in the child. So thermogenin is found in the babies and child, which help to maintain the body temperature of babies. Now time for MCQs. So first question is, well, all of the following statements are true regarding electron transport chain except located in inner mitochondrial membrane, components are organized in decreasing order of redox potential, involved with ATP synthesis, cyanide inhibits electron flow. Correct answer is components are organized in decreasing order of redox potential. Next, a naturally occurring uncoupler is dicomerol, 2,4-DNP, thermogenin, salicylate. Answer is thermogenin. Next, third question is, the oxidation and phosphorylation of intact mitochondria are blocked by puromycin, oligomycin, streptomycin, erythromycin. Answer is oligomycin. Next question is, respiratory chain is found in mitochondria, cytoplasm, nucleus, endoplasmic reticulum. Answer is mitochondria. Next question is, in the oxidation of one molecule of NADH produces 2 ATP molecule, 2.5 ATP molecule, 4 ATP molecule, 1 ATP molecule. Answer is 2.5 ATP molecule. Next question is, which of the following vitamin is involved in electron transport chain? Thiamine, folic acid, riboflavin, cobalamin. Answer is riboflavin. I hope दोस्तों, if you like my videos, please like, comment and subscribe my channel and share with your friends.